Good afternoon, everyone. Welcome to all of you here in Missouri Auditorium and to all of those listening by video conference. And I've already assured our speaker that the number of people who are going to soak up the science that she presents is considerably greater than the people she can see in front of us. But thank you for those of you who have turned out in person, because it's always a little bit more interactive that way. I'm delighted to introduce our speaker, Melanie Cobb, who is a professor of pharmacology at UT Southwestern Medical Center and holds the Jane and Bill Browning Jr. Endowed Chair in Medical Science. If you're watching the calendar, you might know that she originally was scheduled to speak to us on April the 4th. And if you weren't paying attention to what else happened that day, you might wonder what happened to that plan. Well, it was not exactly in her plan not to get on a plane that day, and it wasn't on our plan not to have her arrive here. It had something to do with a tornado, or a whole series of them that plowed through Dallas and rather disrupted the Dallas-Fort Worth airport for quite a few days. So we were very pleased that despite that particular unfortunate occurrence, Melanie was willing to reschedule and come back and speak to us today. May 30th, we arranged a thunderstorm last night, which delayed her plane from landing for 2 hours. So yes, the tradition continues. Hopefully the flight home will be uneventful. Dr. Cobb obtained her undergraduate degree at the University of Chicago, her PhD in biological chemistry at WashU, after postdoctoral training at Mount Sinai and a 3-year period as a research associate at Albert Einstein. She landed a faculty position at UT Southwestern and has essentially been there since that time for most of the remarkable career that she has carried out, studying a variety of issues, but particularly well known for her work on MAP kinases. At UT Southwestern, she for a time was the Haberach Dean of the Southwestern Graduate School, and now, as I've mentioned, is professor and holder of an endowed chair. For all of us who have struggled trying to understand MAP kinases and MAP kinase kinases and MAP kinase kinase kinases, and I don't know how many Ks we're up to now, it's always a relief to have somebody come and explain this stuff. And not only do we have a distinguished speaker, member of the National Academy, to lead us through this, but she has made seminal contributions to this whole field, particularly in the original identification of ERK1 and ERK2 and many other steps that have followed, now being applied, as she will tell us about today, in terms of their function in the pancreatic beta cell, with obvious implications for diabetes, a disease, of course, which is in virtual epidemic proportions right now in our country and increasingly so in the rest of the world. So it is a great pleasure to have her here today. Please join me in welcoming Professor Melanie Cobb.
Thank you very much for the introduction, and thank you for inviting me twice. And I'm glad I actually made it one of those times. Let's see. Okay, so just briefly today I'm going to tell all stories really about pancreatic beta cells after a little bit of background on ERK1 and 2. Okay, so I think it's become increasingly clear that ERK1 and 2 are involved in all kinds of processes in cells, and in fact, I can think of few events that don't have some input from these particular MAP kinases. They're key in proliferation, in cell lineage commitment, and maintenance of differentiated functions, and that's what I'm most interested in and why we've looked a lot at pancreatic beta cells. They're misregulated in all kinds of disease syndromes, particularly cancer, due to activation of upstream activators of the pathway by mutation in a number of cancers. There are a whole range of developmental syndromes that end up impacting the activity state of ERK1 and 2. Some of these are among the most common developmental syndromes in people. And there are also negative regulators of tyrosine kinase signaling that are involved in causing ERK-impacted developmental issues. So a number of years ago, through collaborations with Betsy Goldsmith and one of her postdocs and students who might be here as a fellow, we solved the crystal structures of ERK2 in the unphosphorylated state here and in the phosphorylated state. Looks like a typical protein kinase. One of the nice things about it is it's a relatively small protein, so we could see the whole protein. It's about 41 kilodaltons, and it has two domains, a smaller N-terminal domain and a larger C-terminal domain. And ATP binds here at the interior of the interface between the domains. It's activated by two phosphorylations, and in the case of ERK2, that activation is really profound, causing about a 50,000-fold increase in KCAT. And the two phosphorylations are in this activation loop region here, and they cause structural rearrangements in the protein to cause this activity increase. A couple of points I'll come back to later, a little bit about the MAP kinase insert. These purple regions are regions that are not contained in the core kinase domain. They're inserts in MAP kinases in particular. This insert here and winding up the back of the protein, we have a C-terminal region that has an important 310 helix here and helix at the top of the molecule. In terms of detection of activity, while we used for many years immune complex kinase assays, now just about everybody uses antibodies to the phosphorylated forms of ERK1 and 2. A couple of reasons those are so good. One is that the two phosphorylation sites are only separated by a single residue. And the second reason are these are really rather abundant proteins. As we think of signaling proteins sometimes as perhaps somewhat rare, but these may be on the order of a couple of hundred nanomolar or even a micromolar perhaps in certain cells. So there's a lot of them, so they're easy to measure, to detect. And so I'm going to talk about this pathway, and I just, for the sake of full disclosure, want to point out that ERK2 fits here in this example of a signaling network, and it's very easy to focus on one little pathway and lose sight of the big picture, which is all of these interconnections. And so more and more we're trying to acknowledge these interconnections and figure out how they really contribute to what else happens when this pathway is turned on. Okay. So much of what I'm going to say will focus on specificity in different ways, and the first thing I want to point out is although this cascade can be activated by all kinds of ligands, almost every single agent that affects cell function will change the activity of ERK1 and 2. There is one step where incredible specificity is generated in MAP kinase pathways, and I have three pathways here. This is the MAP kinase step, and just to clarify, this is the MAP2 kinase step, or MAPKK, and it is at this step that we have incredible specificity, and this just shows here phosphorylation and activation of, in this case, phosphorylation of ERK2 by the MEKs in its pathway, the MAP2Ks, compared to the MAP2Ks in the P38 pathway. And you can see there's more or less absolute specificity difference. So while lots of things turn on this pathway, they have to work through this particular cascade to do it. So I mentioned that many agents control this pathway, can change its activity, almost a dizzying number, and it's sometimes hard to get a grip on how different activating mechanisms can lead to different outputs. And what you can see here is old data, about 20 years old, from a whole bunch of different laboratories who've worked on this problem. Activation of ERK1 and 2 in PC12 cells by two different ligands, EGF, which gives a very rapid increase in activity that's extremely transient, and NGF, nerve growth factor, which causes prolonged activation that can even be detected the next day. And so the differences in ligand activation mechanism has a big impact on the output of the pathway, since this pathway is associated primarily with proliferation of PC12 cells and NGF with neurite extension in PC12 cells. So phosphatases have been implicated as at least one mechanism for controlling this type of ligand-dependent kinetic specificity. And this will come up again when we get to beta cells in just a minute. So another important factor in determining what these enzymes do, they can be activated by a number of ligands, but they don't always localize in active form at the same place. And I'll show a couple more slides about that, but first I want to point out there are a large number of scaffolding proteins that direct these enzymes, localize them in particular places depending very much on which ligands have activated them. So for example, ones that we're particularly interested in are these nuclear pore proteins, and at least 3 or 4 of these proteins are known to be very tight binders for MAP kinases. There are some proteins like PEA15, which keeps ERK2 out of the nucleus. There are scaffolds that localize to the cytoskeleton and to various organelles, for example, and ones that have very distinct cell type dependent functions, such as these KSR proteins. So scaffolding has a lot to do with it, and actually this may be one of the least understood aspects of how these pathways are controlled. So about location. We started looking at nuclear localization a number of years ago, quite a long time ago, and what we found was if we used recombinant ERK2 and permeabilized cells, digitonin permeabilized cells, we could see that the protein localized in the nucleus, whether or not we added an energy source or cytosolic transport factors. And here's a study with GFP-ERK2, which you can see also localizes to the nucleus. In this case, we compared its nuclear localization in these reconstitution studies to NLS-BSA, an import substrate used frequently to look at beta-1 dependent import. And you can see here that import of NLS-BSA requires transport factors, and it requires an energy source because A-pyrase eliminates its nuclear import, whereas ERK2 gets into the nucleus equally, pretty much regardless of additional factors added. And in fact, import carriers compete with ERK2 for entry, which is because ERK2 binds to a similar location on nuclear pore proteins as the import factors. And so here, for example, is a schematic model of nuclear pore proteins on the nuclear pore. And some of these proteins contain a whole string of FX-FG type motifs. And these were shown by Kerry Kornfeld's lab to interact with MAP kinases, in particular ERK1 and 2, with some selectivity. And so ERK can bind to these FX-FG motifs that are common on these nuclear pore proteins. And you can see here just a pull-down assay showing binding of ERK2 to the C-terminal domain of nucleoporin 153. And so 358, nuclear pore protein 358, and TPR have also been shown to bind. And I believe there are at least 2 or 3 others that have also been shown to bind. So ERK2 probably spends a lot of time around the nuclear pore. Interestingly, most of the characterization of the localization of ERK2 was done in transformed cells, the standard cell lines that people use in the laboratory, like Ref52 cells, for example. And here, if we use antibodies to phosphorylated ERK2, you can see in the unstimulated state, there might be a little bit of a signal from one cell. But if you look at cells stimulated with either EGF or 4-bol ester for 10 minutes, that much of the protein is localized in the nucleus, although you can see some localization around the cell periphery in the case of EGF. On the other hand, if you look at untransformed cells, cells that were instead immortalized but not transformed, like human foreskin fibroblasts, and we've looked at a lot of other cells that have been immortalized like this, what you see is something somewhat different. Stimulation with EGF doesn't cause that pronounced nuclear localization, whereas stimulation with 4-bol ester does. So I think in non-transformed cells, which haven't been the models that we've used very much, but by and large in non-transformed cells, the localization of these enzymes can be quite different depending on the activating ligand. Okay, so that's all the background. The rest of what I want to tell you focuses on the role of these enzymes, ERK1 and 2, in pancreatic beta cells. One of the reasons, again, that we were quite interested in their actions in beta cells is that we found early on that they were important for regulating insulin gene transcription, and I'll have a good bit more to say about that. One of the fascinating things about beta cells that we've learned is that ERK1 and 2 are activated by all of the signals that stimulate insulin secretion, and they're inhibited by agents that prevent insulin secretion. So they're actually really wonderful markers of stimulation of beta cells to productive insulin release. I don't think they're directly involved in secretion, or if they are, it might be a more minor role than some of its other roles. Nevertheless, they're wonderful as a readout of stimulatory agents on beta cells. And so we've used it and learned a lot about how beta cells work using this model. So just to show some pictures of beta cells, and one of my points here is that this is a colored in model with alpha cells in red and beta cells in green. And you can see here is a mouse islet from a normal mouse, and here is a human islet from a relatively normal human. And one of the things that you see is the ratios of alpha and beta cells are quite different. And the idea that all of the alpha cells are located on the outside of the islet doesn't really hold up when you look at normal human islets so much. And so while we use human islets for all of the studies that we can to look at signaling, we try to reproduce everything in human islets. We've used beta cell lines a good bit. I think the point here is that mice might not be the perfect model to study control of pancreatic beta cell function, just this architecture issue alone is important. Okay, so I'll show this slide twice. So the things I want to really point out is that all of these agents that activate insulin secretion, amino acids, glucose, of course, acetylcholine, GLP-1, all of them activate ERK1 and 2, and they all do it through calcium influx, and they all require calcineurin, the calcium-dependent phosphatase, to be able to activate ERK1 and 2. Agents such as epinephrine, which acts through the alpha-2 adrenergic receptor, turns on GI, and GI blocks insulin secretion, and it blocks ERK1-2 activation by all of these agents. So all of these nutrient sensing events depend on calcineurin. Agents that activate ERK1 and 2 through more conventional tyrosine kinase ligand type mechanisms, they don't require calcineurin. They can activate ERK1 and 2 whether calcineurin is present or inhibited. So somehow there's an insulation between the glucose nutrient sensing pathways and the typical pathways that we think of as regulators of ERK1 and 2 and most other cell types. Okay, so first, some evidence for what I'm telling you about this nutrient sensing and insulin demand selectivity. So here is a comparison of ERK1 and 2 activation. Here's the total amount of ERK in the lysates of these cells, and then a measure of activity using antibodies to phospho-ERK1 and 2. And you can see at either 11 or even 3 millimolar glucose, which is pretty low. 11 is pretty high glucose concentration. ERK1-2 shows some activity, although it's clearly increased at 11 relative to 3. If we add forskolin as a cyclic AMP increaser, which is more or less a mimic of GLP-1, you can see we get a potentiation of activation. When we've looked at this type of concentration curve in more detail, what we see is ERK1 and 2 are activated almost maximally by about 8 or 9 millimolar glucose. And resting concentration, we've now been using about 4 or 4.5 millimolar glucose, since that's the new standard for a normal glucose tolerance concentration. You can see here we still see some activation at 3. So I guess the important point here is that ERK1 and 2 are activated in beta cells under all normal conditions. The amount of activity changes, but it goes up and down over a fairly small range. One reason why we can't do all the experiments we've done in islets is the following. Here's what we see if we look at alpha cells, and both of these are cultured cell lines. If we look at alpha cells, what you see is ERK1 and 2 are low at high glucose and high at low glucose. So it's just the opposite of what happens in beta cells. So alpha cells work an opposite way. So if we look at whole islets, you can imagine that the signal that we would see from beta cells will be mitigated somewhat, reduced a little bit by the or maybe a lot, by the contribution from alpha cells. And here you can see an activation, very nice activation by insulin in alpha cells. Okay, so alpha and beta cells don't work the same way, and if we want to understand the signaling, we're somewhat restricted to using beta cell preparations or isolated beta cells from intact islets, which we've done on numerous occasions. And when I say intact islets, all of the work we've done on intact islets that I'm showing you here was done with human islets from transplant facilities. Okay, so the issue of nutrients and secretagogues activating ERK1/2 by mechanisms different from growth factors. So here's some evidence to support that. And it's important to point out that FK506 is an inhibitor of calcineurin. U0126 inhibits MEK1 and 2. So basically this is a molecule that will prevent ERK activation, and this one will dampen calcineurin activity. So here you can see glucose activation of ERK1 and 2. It's very nice. If we add calcineurin blockade to that, what we see is a significant suppression of ERK1/2 activation by glucose. If you look over here at GLP-1, you see something pretty similar, stimulation of ERK1 and 2 with GLP-1 and a blockade with calcineurin inhibition. So very clear. And this is true again with all of the agents that enhance insulin secretion, all of the nutrients and hormones. Okay, growth factors that activate ERK1 and 2 in almost any type of cell you'd look at. Here you can see formyl ester activating ERK1 and 2, but no blockade by blocking calcineurin, so no requirement for calcineurin to get the pathway turned on. And here's EGF, again, nice activation of ERK1 and 2, but no blockade by calcineurin. So nutrients and growth factors activate this pathway by distinct mechanisms, even though we presume they use all of the same kinase cascade that everyone thinks about for controlling the pathway. Okay, what about secretion inhibitors? I mentioned before that epinephrine blocks insulin secretion from beta cells, and that happens through alpha-2 adrenergic receptors, and you can see here is activation of ERK1 and 2 by glucose. If we include an alpha-2 selective agonist, we block activation of ERK1 and 2, and if we use epinephrine itself, we also block activation of ERK1 and 2. For comparison, if we look at a HeLa cell, a typical cell where people have characterized how some of these pathways work, you can see that control conditions, there's no activity detectable. If we add epinephrine, we can activate ERK1 and 2, not inhibit it, but turn it on. If we add this UK, the selective alpha-2 adrenergic agonist, you can see we can also activate ERK1 and 2. So turning on GI in an average cell that's used for this kind of experiment will activate ERK1 and 2. In beta cells, it completely turns it off. And again, the growth factor insulation component of this argument, you can see here that glucose again is blocked by the alpha-2 adrenergic agonist, but there's absolutely no effect of that drug on NGF activation of the pathway. So just putting that all together, we have all these nutrient sensing mechanisms, all of these mechanisms that are telling the cell that more insulin needs to be secreted. They all require calcineurin in order to turn on this pathway. And this agent, which will inhibit insulin secretion, blocks ERK1/2 activation. So one of the questions that really puzzled us for a long period of time was how the phosphatase calcineurin is required to turn on this kinase cascade. Why? What's it doing? And what I'll show you is some evidence that it's actually working by dephosphorylating B-Raf, which is the MAPK or MAP3K upstream of this cascade. Okay, and all of this work was done by a former graduate student, Ling Ling. And the first thing she had to do was figure out what the MAP3K was upstream of the pathway. There are multiple RAF isoforms. RAF is generally considered to be the MAP3K upstream of ERK1 and 2. And so she assayed all three of these isoforms. She couldn't measure any activity from ARAF, and here is what she got with CRAF or RAF1. You can see basically no activity detected. Just to show you she could really do the assay, here's a PMA-stimulated HeLa cell with the same type of CRAF immune complex kinase assay, phosphorylating its kinase dead substrate here, and you can see the assay works fine. On the other hand, if she immunoprecipitated B-Raf, what she found was activation by glucose and reduction in activity by calcineurin. So this is just a confirmatory, the expectation was that CRAF would be the activator. It's the most commonly considered activator in a typical cell for the ERK1 and 2 pathway. And so what Ling did was to immunoprecipitate C-Raf and then show that she could bring down the protein by blotting it, and then also blot with an antibody to phosphoserine 338, which is a key activating phosphorylation site on the protein. So phosphorylation of this site would indicate that it had been activated, but in fact, if anything, the phosphate content decreased following exposure to glucose. Okay, so another experiment here, she did a time course of stimulation with glucose, B-Raf IP kinase assay again, and if she treated with calcineurin, cyclosporin A, another inhibitor of calcineurin, she could reduce that B-Raf kinase activity. So that seemed pretty straightforward, but then she did another experiment based on a lot of work from a number of laboratories, initially from Joe Averick's lab, but a huge number of groups have really presented incredibly valuable information that shows how complicated this kinase pathway can be. And that involves the idea that these RAF isoforms can dimerize, heterodimerize, and that the partners in the heterodimer combination can both positively and negatively influence each other's activity. So just to see if that was going on in beta cells, what she did was she IP'd C-Raf and then she blotted for B-Raf. And so here's her C-Raf blot, so you can see it was present either in untreated cells or cells stimulated with glucose. And here's her B-Raf blot. It did not co-precipitate with C-Raf in the unstimulated cells, but it did in the stimulated cells. So we do have the issue of combinations of these molecules being present in some of the immunoprecipitates and possibly affecting each other's activity. So to answer that question finally, what she did was to use RNAi in beta cells, and this is not such an easy prospect in beta cells because just about the best knockdowns that we've achieved in these cells is about 50 to maybe 60%. And so here what she did was knock down B-Raf, and you can see ERK activation is attenuated. This is her B-Raf blot. So this is the reduction from this to this with B-Raf knockdown, and here's just a plot of the activity. Here's C-Raf, and this one was a bit of a surprise. She stimulated, and when she knocked down C-Raf, she actually got an increase in ERK1/2 activity. So short term, what that suggests, which is consistent at least with other data in the literature, is that C-Raf could actually suppress the activity of B-Raf in a relatively rapid assay. Interestingly, when she looked at a couple of hours of stimulation with glucose, she found the opposite. In this case, C-Raf was activating and forcing more activity through B-Raf. So a complex, time-dependent response of these kinases to activation by nutrients. Okay, so how was calcineurin in this picture? What was it doing? And what she figured out was that calcineurin essentially dephosphorylates a negative feedback site on B-Raf, and that site needs to be dephosphorylated in order to allow continued activation of B-Raf. If the site's phosphorylated and that site isn't reversed, basically, B-Raf will be turned off and ERK can't be activated any further. And so a couple of different experiments to put that idea together, some of which depended on determination of B-Raf phosphorylation sites by Debbie Morrison's lab a couple of years ago. So here you can see she's stimulated with glucose, and here she's assaying B-Raf activity using phosphorylation of kinase dead MEK1 again. And there's the increase in activity. If she blocks calcineurin, the activity is decreased. So we know B-Raf is the sensitive step, or at least one of the sensitive steps. If she blocks ERK activity, she does not get this, she still can measure B-Raf activity, and if she inhibits calcineurin and blocks ERK activity with the MEK inhibitor, she doesn't suppress B-Raf activity any longer. So by blocking ERK, she's preventing the feedback inhibition. And here, you can read the red lines if you couldn't follow what I said, because it's fairly confusing. So she identified a site that seemed to carry most of this inhibitory activity, and that was threonine 401. And so she then expressed B-Raf in cells and compared the wild-type protein to the threonine 401 alanine mutation. And so here you see stimulation by glucose and depression of activity by blocking calcineurin. With T401 B-Raf. She found a stimulation of activity, actually higher basal activity to begin with, and there was no suppression by inhibiting calcineurin, presumably because this site can no longer be phosphorylated, so it doesn't need to be dephosphorylated. So altogether, that gives us this model, which is calcium influx enhances calcineurin activity, and its important function is to dephosphorylate B-Raf so that it can continue to lead to activation of ERK1 and 2 by nutrients. Okay, so next, a few slides on what we've done to look at ERK1 and 2 and regulation of insulin gene transcription, and we've worked on this for a long time, but a lot of the work was done by a former graduate student, Kushi, and then much of it has been done by a former postdoc, Michael Lawrence. And this was the key experiment that Michael did that really made it important. Every detail of how we did these experiments became important. And so what he showed here, it was well known that glucose could suppress insulin gene transcription, but we didn't know anything about the time course. So what he did was to show that glucose activated the insulin gene promoter on the short term. This is 2 and 6 hours. By 12 hours, though, he found a suppression of that stimulation. And by 24 hours and thereafter, he found suppression below basal transcription level. Okay, so that wasn't too surprising. The surprising part was that if we blocked ERK1 and 2 activity, we did reduce insulin gene transcription in response to glucose, so we're basically close to basal levels at this point. But if he blocked ERK1 and 2, he restored the suppression caused by high glucose at long times. So we're looking at a biphasic effect of glucose, and we're looking at a multiphasic effect of ERK phosphorylation. If we block ERK activity early, we prevent a stimulatory effect on the promoter. If we block ERK activity late, we prevent the inhibitory effect of glucose on the insulin gene promoter. And so this became really a key to how we did many of the experiments that come next. So we're really basically comparing rapid or somewhat acute stimulation with glucose to a more chronic long-term stimulation. And this just shows the first experiment that we did in collaboration with Mike German and Steve Griffin, then at Mike's still at UCSF. It's a cat reporter assay, and in this case, it was in freshly isolated mouse islets. And here at low glucose, you can see relatively low promoter activity, whether or not ERK is added, is transfected. When he's stimulated with 16 millimolar glucose, you can see an increase in this promoter activity. And when he blocked ERK1 and 2, either with another MEK inhibitor or including kinase dead ERK2, he pretty much potently blocked activity from this promoter in islets. And so over a number of years, we and many other people have identified substrates that are transcription factors that work on the insulin gene promoter. And of course, the important factors that have been identified are neuro-D1, PDX, and MAPK. And interestingly, 2 of these, in particular neuro-D1 and to some extent PDX1, are important ERK1/2 substrates. And in fact, neuro-D1 really requires ERK phosphorylation in beta cells to function well. ERK also phosphorylates P300, that's been shown in the literature. It phosphorylates the common general EBOX dimer partner for neuro-D1, E47. It also phosphorylates CEBP beta, which is an inhibitory regulator of insulin gene transcription, and that's been shown for a number of years, for example, by work from Habner's lab. And it can phosphorylate c-Jun. And so we've tried to figure out how all of these factors interplay with ERK signaling to regulate the promoter. And one of the important things that is not an issue if you're working on islets from animals, but it's a significant issue if you're looking at cultured islet cell lines. And that is the maintenance conditions for these cells. Generally, the cells are maintained in very high glucose concentrations. And we wondered why that was, and so Michael lowered the concentration to what would be considered a somewhat more normal glucose concentration. And what he found was after a couple of days, he lost expression of MAPK unless he kept the cells in very high glucose. And inhibiting ERK activity with UO126, the MEK inhibitor, doesn't do anything to this. So the glucose concentration itself has a big impact, even on a relatively short term, on expression of MAPK. We know that Jun expression goes up with low glucose, you can see here, and of course, again, work by Habner and a number of other groups have shown that CEBP beta is induced when cells are exposed to very high glucose for a long period of time. So you can see here, 48 hours on high glucose is enough to induce CEBP beta. So it's important to treat these cells in a particular way so you know what the state of the relevant factors is that will be controlling promoter activity. Okay, so in this case what we did was to look at what happens to factors that we know are important, MAPK, NeuroD1, PDX1, and also CEBP beta. And these are chromatin immunoprecipitation assays using P32 labeled CTP, so you can actually see bands. And these are cells treated for 30 minutes with glucose, and compared to the control, you can see increases in binding of MAPK, NeuroD, and PDX to the insulin gene promoter. And if we use either the MEK inhibitor or the calcineurin inhibitor, we block those interactions. And they're not complete blockades, but substantial reductions. Rapamycin, on the other hand, an inhibitor of mTORC1 primarily, doesn't do anything to these interactions in beta cells. If we depolarize with potassium, we can see binding of NFAT and MAPK, and again, both are blocked by inhibiting ERK1/2 activity. If we look at cells for comparison that have been incubated for a couple of days in high glucose without any kind of removal of that glucose, what you see here is that dropping the glucose for 30 minutes and then adding 11 millimolar glucose, we don't see any binding of MAPK, NeuroD1, or PDX, but we do see binding of CEBP beta. And this is partly because the CEBP beta concentration is much higher in cells in 11 millimolar glucose. Interestingly, if we treat these same cells with potassium depolarization, we can still see binding of NFAT and MAF. So it isn't just that CEBP beta is preventing interaction of these other factors. Okay, so one of the observations that we made that's gotten us particularly interested in studying this in more detail is what happens with the signaling molecules themselves. So here we did chromatin immunoprecipitation, and this was on human islets with antibodies to ERK1 and 2, antibodies to its downstream substrate RISC2, which can phosphorylate histone tails, NFAT, calcineurin, again the phosphatase. And you can see that we can see ERK1/2 RISC and calcineurin associated with the insulin gene promoter under stimulatory conditions. And likewise, we can see here with GLP-1, but if we add FK or UO126, the interaction is reduced. And we can see that under a number of different conditions and with other promoters. And so we're now exploring the density of interactions of ERK1 and 2 with chromatin and beta cells. Just for comparison, again, my point about ligands doing different things, if I haven't made that strongly enough yet. Here again we have Min6 cells, and we're comparing glucose, EGF, and NGF as activators of these cells, and looking at activated ERK1 and 2 by immunoblotting. And if we block MEK, you can see that we can eliminate some of the signals or reduce them quite substantially. Here now we're also looking at CHIP, chromatin immunoprecipitation, and you can see that we're comparing the insulin, the FOS promoters, and glucose and EGF don't activate the FOS promoter very much in beta cells, but NGF does, and you can see a big difference in interactions. And if we add UO126, which again blocks ERK1/2 activity, you can see we can dampen the glucose-dependent interaction consistent with the CHIPs I was showing you before. We don't really inhibit EGF signaling very well in these cells, and you can still see a residual ERK1/2 activity, and you can see a reduction in both types of interactions in response to NGF. So different ligands do different things, and that includes different DNA associations with activated molecules downstream of those ligands. So one of the things that we noticed was that ERK1 and 2 had an impact on histone acetylation in beta cells, and this is to show you not so much about the histone modifications themselves, but about the glucose dependence of these interactions. So here, if we look under normal conditions, you can see at stimulatory glucose an increase in acetylated H3 and H4, and an increase in binding of P300 to the insulin gene promoter. If we block ERK1/2 activity, all of these are reduced, as you can see here. If we look at cells that have been exposed to high glucose for a couple of days, the pattern is quite different. You can see here that under low glucose conditions now, we can see association of P300 and acetylation of histones, and blocking ERK1/2 at high glucose enhances their association relative to glucose alone. And this is basically a comparable type of experiment. Here we're looking at cytokine action, and I'll get back to that in a few minutes, in just a minute, but the combination of high glucose with IL-1 beta cytokine causes basically an inhibition of acetylation of histones, of binding of P300 and binding of polymerase II to the insulin gene promoter. But if we block ERK1 and 2 under these conditions, so this is an exacerbated chronic glucose situation, exacerbated with the cytokine, then we no longer lose the interaction by blocking ERK1/2 activities. And this is more of the same. This is in human islets. Again, preincubated in a resting glucose concentration, we see pretty normal behavior with stimulation of histone acetylation as we add glucose. But if we incubate these islets in high glucose for 1, 2, or 3 days, we get the same kind of reversal that I was showing you before. Instead of increased acetylation as we increase glucose concentration, we see decreased acetylation. And so basically what we're seeing here is that the condition of the beta cell, not the activation of the kinase pathway, is a major determinant of what's on the promoter and how the promoter can be regulated by the signaling pathways. Okay, and this might be the most obvious set of experiments showing that. So here we looked in more detail at what happens when cells are treated with either resting or stimulatory glucose under chronic conditions along with IL-1 beta. And so ERK1 and 2 is activated by glucose, but it's also activated by the cytokine, perhaps a little bit differently under each condition, but basically all of these conditions will cause ERK activation. The stress-associated MAP kinases, P38 and JNK, or SAPK, another name for it, these are activated just by the cytokines. So you can see here addition of the cytokine, completely independent of the glucose concentration, activates these protein kinases, although ERK is pretty much activated under all these conditions. So now what happens when we activate all these pathways? So these are time courses, again, chromatin immunoprecipitation. And what you can see here is that ERK1/2 bind right away and are still associated even under this condition, which I'll show you in a moment, has turned off this promoter completely. P38 likewise, JNK likewise, RISC likewise, calcineurin likewise. So all of the signaling molecules bind right away when they're stimulated and active, and they stay there. Okay, what about factors that are important for insulin gene transcription? You can see MAPK initially binds, but in the presence of the cytokine along with the high glucose, it's gone after by about an hour. NeuroD does stay on, PDX1 also drops off. P300 and polymerase are also gone. So one of the things that happens, it takes about a day of high glucose to induce CEBP beta, but if we add IL-1 beta, we can see an increase in this molecule actually by a couple of hours. Here's a 4-hour time point. So it's a much faster induction of this inhibitory transcription factor. If we did nuclear run-on at 4 and 24 hours just to see what was actually happening to transcription from the insulin gene promoter, and at 4 hours, in the presence of the cytokine, but at low glucose, a moderate amount of glucose, you can see we still had transcription initiation ongoing, so we could see nuclear run-on. But if we looked at 16 millimolar glucose at this 4-hour time point, which corresponds to this last column, you can see a suppression of transcription. There wasn't any ongoing initiation, so there was no nuclear run-on that we could measure. So I guess the main point here is that the kinases may still be bound to the promoter, and they're regulated the same way regardless of the conditions in the beta cell. So if you lower glucose, ERK1/2 activity will go down. If you increase glucose, ERK1/2 activity will go up. But if the cells are treated under circumstances that promotes their their resistance, their glucose insensitivity, then what ERK does in the signaling standpoint, from the signaling standpoint, will not be the same because the protein complement in these cells is different. Okay, so a point of great interest to me, but not necessarily immediately relevant to beta cell function, is what's the molecular basis for ERK1-2 chromatin association, and we're working hard to try to understand this better. First, of course, ERK1-2 bind all kinds of DNA-associated factors. They bind a lot of HLH proteins, ternary complex factors, P300, many other nuclear binding proteins. They also bind nuclear structural proteins, such as the nucleoporins that I mentioned before. They affect a whole group of histone modifications through regulating RISC and MSK, another histone tail kinase. And it was reported about 3 years ago now that unphosphorylated and phosphorylated ERK2 can bind directly to DNA. We find that phosphorylated ERK2 is a better DNA binder, but we also see direct binding to a number of different regions of DNA. And so we're exploring what that might mean. And one idea is that it may occur through basic residues in the MAP kinase insert that was suggested by this paper, but also before that by a paper from Maria Schumacher, who determined the crystal structure of a bacterial kinase that has a eukaryotic kinase fold associated with bacterial DNA. And what she found is this is the part of the kinase, the structure of the kinase looks just like this. This is the part of the kinase that was touching the DNA directly. And this is the MAP kinase insert, which is unique to this particular branch of the kinome. Okay, so far we've done a lot of chip sequencing, and we have identified a number of interactions in beta cells. We do find that there are a number of interactions that overlap with P300 binding sites, so this may account for some of the interactions we can detect. Okay, so overall, ERK1 and 2 and transcription. MAPK activities reflect incoming signals. They're on if they've been stimulated to be on, and they're not if they're not. They're present widely on chromatin and in transcription complexes. The responses they generate depend a lot on what's there, and changing glucose concentration in beta cells alters the profile of transcription factors present among many other proteins that change. So ERK1 and 2 regulate context-dependent histone modifications and other kinds of transcriptional responses, and they're important for insulin production in beta cells. Okay, and briefly for the last couple of minutes, I'd like to tell you about some studies that have been carried out by a postdoctoral fellow, Alaj Jum, in the lab. And the results of these experiments were actually quite shocking to me, very gratifying, and I hope we can turn them into something a lot more significant even than they might appear here. And so this all began as a collaboration with somebody in cardiology, Professor Jay Schneider, who did a chemical screen about 10 years ago almost for molecules that enhanced cardiac differentiation using a reporter assay. And he found a whole group of compounds and started looking at how some of them work. And one of the things he noticed is that the compounds could increase expression of neuro-D in neuronal cells. And so we thought it would be a good idea to look, since neuro-D really, it's really beta-2, it masquerades as neuro-D. So we tried these compounds on islets, and we get as many islets, human islets, as we can handle, and when we can't use them all acutely for experiments, we put them in the incubator. And when he got this compound to work on, we actually had islets that had been in the incubator for a year, human islets just sitting there in the incubator. And at this point, they don't make a lot of insulin, and they don't secrete any in response to glucose, although they might secrete just a little. And so he started looking at them, and what he found was, and this happens to be a 6-month-old set of islets, what he found is within 2 days, insulin mRNA went up by more than 60-fold in this experiment by treating with this compound. And that was pretty shocking. Interestingly, glucagon went down several-fold. He looked at insulin secretion, and you can see here by ELISA, the compound increased the amount of insulin protein secreted quite substantially. And so a question is really, this is an increase, but is it significant? How does it compare to what a fresh islet would secrete? And so he compared islets that had been in the incubator this time for 3 months with ones that we'd had fresh, basically, from the transplant facility at UAB. And he either treated them with the drug in the light bars or not. And then he compared insulin content. So this is protein, not mRNA. And you can see here, this is the amount in the fresh islets. And after treating the old islets with the drug for, I think, 2 days, you can see that we're at least within the right order of magnitude for insulin production from fresh islets. Furthermore, the compound increased insulin content of the fresh islets after only 2 days. So this was a really amazing result to me. So he's analyzed the actions of this compound in a number of ways, and he's looked at a lot of changes in gene expression. And we start out with the 3 genes that are essential for insulin gene transcription, neuro-D/beta-2, PDX1, and MAPK, and you can see they're all increased as a function of time after exposure to the compound. That was really good, but more surprising still was after, again, a time course of treatment with the compound. He looked at GLU2 and glucokinase, so the glucose sensing apparatus basically of beta cells, and you can see they're both increased a lot. This is a really huge increase in glucokinase expression. So then he sort of stood back and looked at all of the genes that are involved in the beta cell differentiation pathway, and they're listed here, and more or less in sequence of their expression during beta cell differentiation. And he actually looked at everything on here, and some of them are shown here. A lot of them show up in multiple of these states, of course. And you can see here huge increases in NKX 6.1 and NKX 2.2, also huge increases in neurogenin 3 and PAX4. And interestingly enough, these 2 factors of all of these are not as highly expressed in mature islets as some of the others, and they went up very tremendously right at the beginning and then began to drop slightly following further time of treatment with the compound. And so everything he looked at basically increased with time of treatment with this drug. And so he did look a little bit at histone acetylation and some signaling in these cells following drug treatment, and this is a time course. And I want to point out that this time course looks quite different from what I showed you with glucose or G-protein coupled receptors in beta cells, where we saw acute responses within the first few minutes. You can see a little bit of activation of ERK2 here at 15 minutes, but tremendous activation at about 4 hours that's somewhat persistent, although drops somewhat. This looks a lot more like an NGF type response, although it's not very pronounced early, so it's quite delayed. Doesn't really look like any obvious signaling molecule to me. You can see a time-dependent increase in acetylation of H3 and H4, and here, not such a big change in H3K27 trimethylation. And so this is Min6 mouse beta cells, and here we have human islets, and you can see to the extent that he looked, these changes are actually quite similar. Interestingly, one idea was that this compound would affect histone acetyltransferase activity, but in actual fact there was no effect on one idea that everybody expected was that it would affect HDAC activity, but it didn't. In fact, it did increase HAT activity. This is just treatment with a drug compared to TSA. And you can see a big increase in HAT activity and nuclear extracts from the cells. And when he reconstituted P300 and looked at effects of ERK on its activity as well as the drug, you can see the drug increased activity relative to untreated, and that ERK1 and 2 did somewhat suppress activity compared to the drug treated without ERK inhibition. So we have compounds that seem to really enhance beta cell function. They improve islet function in culture and islet transplant efficacy in diabetic mice. We haven't done enough animals yet, treated them to have statistical significance, but it seems very likely, based on our current data, that the drug can be used to treat animals and increase their glucose tolerance. So what we don't know at this point are what are the mechanisms of action, and we're looking at this very intensely. And we'd also like to know how long the effects are stable. If we take the drug away, do the effects stop? once we remove the drug, how long will beta cell function remain improved? And finally, we're looking at similar compounds that seem to work at lower concentrations. We were looking at 10 and 20 micromolar compound. We'd like to be looking at nanomolar concentrations of similar compounds to see if they improve type 2 diabetes or perhaps rejuvenate islets for transplantation or stabilize them for transplantation. So, and just all of this came from an interest in ERK signaling in beta cells, and we're hoping it'll go a lot farther than that. And so people who did the work, Elaj wasn't at our Christmas party this year, but here he is, and a lot of people in the lab have contributed, in particular Kathy, who's actually maintained islets in culture for us for about 10 years. Thank you very much.
So thanks very much. We have time for some questions. If people would like to pose them, please use the microphones in the aisles so that individuals watching by video can also hear. While people are thinking, this very interesting isoxazole story you're telling, and it certainly raises the question of what's the earliest response that happens when you add the compound. You showed us a variety of transcripts that seem to be going up fairly dramatically. Have you basically done a complete global analysis these days, I guess, by RNA-seq, or at least by microarray, to see what happens in the very earliest moments after exposure and see whether you can identify what might be driving this in terms of the primary effect?
No, we haven't done that. We've certainly done some initial microarray experiments, so we haven't really done anything global or quantitative, and we haven't looked at early times. What we've tried to do is identify direct targets by affinity chromatography, and we have some candidates, but I'm somewhat skeptical so far because I'd like compounds that bind a lot better than, or more efficacious before we, more potent before we really get serious about what potential targets might be. But I think that's really our goals at this point. Initially are to show efficacy in animals and try to identify direct binders.
Yep.
Thank you for a comprehensive coverage of the kinases involved. So the question is, we understand quite a lot about the signaling pathways, so why there are so few drugs for type 2 diabetes? Besides metformin, I'm a diabetic and so interested.
Why are there so few drugs for type 2 diabetes?
And the ones which are developed also cause cardiovascular complications, and they are withdrawn, some of them.
And complications. I'll tell you one reason why I think there are so few drugs. It's because we don't really understand enough about beta cells. I had preconceived notions about how these cells would be regulated, and they're incredibly fascinating, but they didn't exactly match my preconceived notions in a number of ways. So they don't have properties that exactly match anything else that's been well characterized, and there aren't exactly a lot of them. So, other than looking at these cultured cell models from rodents, it's quite difficult to look at beta cell regulation, particularly because the other cells in the islet don't work the same way. So I think we just plain old don't know enough. And the more we know, the better off we'll be. But I think understanding more about receptors on beta cells is very important, and learning how to manipulate those receptors so we don't desensitize the cells. One of the things that is quite striking about beta cells is you treat a beta cell for 10 minutes with something and the effect can be very good. You treat for an hour and it'll start to be bad, not good. So they get desensitized or rewired or whatever, however you like to think of it, very quickly, which means that prolonged therapies may be very difficult to achieve. And that's not a drug company concept to look for short-acting agonists. Right?
And a short question regarding the kinase inhibitors. Are they too non-specific to have any effect?
I didn't understand that.
Are the kinase inhibitor, there are quite a few kinase inhibitors. Are they non-specific?
Okay, about kinase inhibitors. I think one of the major things I've learned is that kinase inhibitors probably aren't a good idea here because the signaling is very responsive to what's outside, but beta cells change depending upon what's outside. So you can grow beta cells in 25 millimolar glucose, take it away for 15 minutes and add glucose back, and ERK will be turned on. But the beta cell will have a lot of negative regulatory factors, and they won't have any, any of the things, not any, some of the things they need in order to control what they do properly. So under those conditions, only bad stuff will happen. Inhibiting the kinases may or may not make any difference. So I think what I've learned is the glucose regulation of what's in the cell and the kinase regulation are completely distinct. ERK1 and 2 regulate insulin gene transcription, but they don't control the complement of factors expressed in the beta cell that can regulate any of these transcriptional events. So,
that's kind of related to my question. So you saw with long-term glucose treatment, you see the change in transcription factor profile. So if you treat with your small molecule ISX, do you see an inhibition of that change that you see with long-term glucose? Because it seemed to be kind of the opposite effect with your molecule.
So I will say that we didn't look at CEBP beta. At least I can't recall the data if we did. He probably did, but I don't remember it. But all of the factors that are important to stimulate for stimulatory transcription, they're all induced. And what happens, so these cells are, and the islets too, are maintained in high glucose. So adding the drug still has positive effects even though the cells are maintained in high glucose.
Okay. And then one follow-up, is the small molecule, the effects that you see with that, are they sensitive to calcineurin inhibition, or have you looked at
Okay, only to the extent that ERK activity is contributing to what the small molecule is doing. And it is contributing a little bit. It definitely regulates, ERK definitely regulates P300. And so if you block calcineurin, you'll interfere with the P300 regulation. But I think it's really only partial. ERK is only a contributor, certainly not the major part of the story.
Okay, thank you.
Well, we've reached the 4 o'clock hour, but I encourage all of you who want to continue the conversation to adjourn to the library where FAES has graciously provided us with some refreshments since we, the government, can't pay for them. Let us thank our speaker again for an elegant presentation.